Okay, folks, let's get started. Happy Monday. Wasn't that weather awful yesterday? Terrible. It's supposed to be 80 on Wednesday, so that's what they say. That's what they say. Extra credit? Well, you never know. I do have a weather-related song today because the weather has gotten to me, so you guys will see the song when we get to that. All right. Let's turn our attention. I've been talking mostly about structural things. I spent a fair amount of the lecture on on Friday talking about DNA. I want to say a few things about RNA, and then for the most part, we're going to turn our attention to the replication and recombination and repair of DNA. That's the most of what I'll talk about today. Before I do that, though, I want to say a little bit about RNA. When we talk about DNA, we don't talk about types of DNA. We basically have DNA, or we don't. And DNA, of course, being the genetic material, and RNA performing numerous functions. And those functions are sorted according to the type of RNA that's involved. So you see on the screen a variety of types of RNA that are out there. And the most common ones that we talk about are these three guys right here. Hiccups again. Transfer RNA, which is the smallest of the general types of RNA that we discuss. And it transports amino acids to the site of protein synthesis. So it's the transfer RNAs that carry the amino acids for translation. One of the things that we see with all of the RNAs is that they all have functions related to translation. They all have functions related to translation. The ribosomal RNA plays a catalytic role, that is, in the catalysis of protein synthesis. As we will see later, it's the form that is integral to the ribosome, which is the structure that makes the proteins using the genetic information from the messenger RNA. So the messenger RNA is a copy in ribo form of a portion of one of the strands of DNA. And the portion corresponds to what we call a gene. So a gene is a segment of DNA that codes for protein, and the messenger RNA carries the code of that segment to the ribosome for the purpose of translation. The last three RNAs that you see on here are all interrelated, and they are the most recently discovered. Back when I was a graduate student, back in the dark ages, we knew of these three types, and we sort of knew of this one right here, small nuclear RNA. In fact, if you did much work with nucleic acids, you become very intimately familiar with small nuclear RNAs, because whenever we ran a gel that contained DNA from cells, we saw this little thing at the bottom. There was a little bright spot that corresponded to what we all said was, oh, that's a bunch of junk RNA. Because whenever you isolate DNA, you get RNA, and these little RNAs are probably just pieces of things that have broken, et cetera, et cetera. Well, we know today that those little pieces that we thought had broken and were useless, in fact, had a very big function. And the big function that they played was to help in controlling the expression of genes, that is, the synthesis of proteins using the genetic information. There are two main things that I will briefly discuss of those subcategories of the small nuclear RNAs. And these small interfering RNAs, also called siRNAs, or the microRNAs, both have effects on gene expression in slightly different ways. And these effects that they have can be to actually stop a protein from being made once the process has started. And as I said, I'll talk more about those a little bit later. The role of messenger RNA in transcription, I'm sorry, not the role in transcription, but the role in translation arises from its synthesis in transcription. One of the things that we see when we look at the organization of genes within prokaryotic cells is that genes in prokaryotic cells are clustered according to function. So we see, in this case, genes A, B, and C might all be related to a cellular function or need. So for example, I will talk later in the class about what's called the lac operon. And the lac operon is a segment of genes in E. coli that all are related to the metabolism of lactose. So they all have a common function, and they're all organized together. And as you can see on this image, they're all synthesized on the same messenger RNA. And that will turn out to be important when we discuss that later. Eukaryotes are different from prokaryotes in that eukaryotes do not have genes organized like this. Instead, eukaryotes have genes organized in pieces. We see them all intact here. There's all of gene A there. In this case, we see gene A. Then there's what's called an intron, an intervening sequence. Then there's another portion of gene A. And those pieces later get put together. So whereas we see the prokaryotes have gene clusters, we see eukaryotes have genes in pieces. And those pieces get put together in a process that's called splicing that we'll talk about later. So we do not see gene clusters at all in eukaryotes. That's a very important thing. No clustering of genes. Instead, we see genes in pieces. The structure of tRNA has been described by many people as a cloverleaf-like structure. If we look at this transfer RNA, we can see a couple of things. First of all, this is one strand going all the way through here. We see regions of duplex, but those duplexes arise from complementary base pairs between different portions of the same strand of RNA. That's only one strand that's there. We could think of these base pairing things as facilitating something like we see in proteins with folding. These base pairs help to organize the structure of this transfer RNA. The transfer RNA has a 5' end and a 3' end, just like DNA has a 5' end and a 3' end. And it's on the 3' end, up here where this OH is, that the amino acid that the tRNA carries gets attached. So this guy gets attached to an amino acid. And then the question is, well, which amino acid gets attached? Because there are 20 different amino acids that go into proteins. And it turns out that the cell has a way of identifying a specific sequence down here and linking the appropriate amino acid up here. And that's the root of the genetic code. We'll talk about that later. But suffice it to say that without transfer RNAs, translation would not occur. And further, the genetic code would make no sense. So both of those functions are embedded in a transfer RNA. Okay? The genetic code and the carrying of the amino acids to the translational machinery. RNAs are also well known actually, particularly transfer RNAs, for being chemically modified. It's not completely clear why these modifications occur. It may be to designate, for example, a transfer RNA as different from other RNAs, but other RNAs themselves also get chemically modified sometimes as well. So it's not completely clear what purpose the modification has. You can see some of those, and again, we're not going to worry about structure there, and since I haven't named them, I'm not going to ask you to come up with the names either. The ribosomal RNAs are the biggest and the most complicated of all of the RNAs. The ribosomal RNAs are found in the ribosome, as I said earlier, and we're so zoomed out you can't even see individual nucleotides in here, but suffice it to say that every one of those little bars right there is a base pair that's occurring between a nucleotide on the one side and another nucleotide on the other side. Okay? So we see that there's a lot of interactions that would correspond to tertiary structure within this ribosomal RNA. And another noteworthy feature of this RNA is it also is a single strand. There's the 5' end right there, and I don't see the 3' end marked. I want to say it's over here. There it is. Okay, yeah, all right. I wanted to say it was like here. Okay. right there is the 3' end. Now, the elaborate structure of these ribosomal RNAs play roles in translation, and we know some of them, but not all of them. One of the main functions of ribosomal RNAs is to catalyze the synthesis of the peptide bond. One of the main functions of the ribosomal RNAs is to catalyze the synthesis of the peptide bond. Now, that's relatively new information, relatively new in that we've known it now for less than about 20 years. Okay? The peptide bond, all right, is the fundamental unit joining amino acids in a protein. And catalysis, before about 30 years ago, was something that we thought was only possible with proteins. But now we know that there are many different RNAs that can actually catalyze reactions, and we call those RNAs ribozymes. It's an unfortunate choice of name because it sounds like ribosomes. Ribosomes are the complexes that make proteins. Ribozymes are catalytic RNAs. Don't confuse those two. Ribosomes contain ribozymes. Okay? Ribosomes contain ribozymes. Now, some other things associated with the elaborate structure of ribosomal RNAs may be the ability of the ribosomal RNA to provide a scaffolding onto which ribosomal proteins bind. So a ribosome contains both RNA and it contains many proteins. And we think that that structure that the ribosome has is inherent to its function. No surprise, you've heard this over and over. And that structures like this may help to properly position the many proteins. There's about 50 proteins found within a ribosome. Okay? That's one of the thoughts about the structure of a ribosomal RNA. Now, small nuclear RNAs are RNAs, as I said, that are relatively recently discovered, and they are involved in processes whereby the process of translation is actually affected. Now, they're called small nuclear RNAs because they typically are very small pieces. Okay? They start as a bigger piece that gets processed down to something that's about 20 to 25 nucleotides long, and it looks like this double-stranded guy right here. Okay? These are double-stranded RNAs. Double-stranded RNAs. And the way that they work, I'm not going to take you through this whole process because I think there's just too much information for us to go through there. But the way that they work is this double-stranded RNA, the cell sees as a warning. It sees it as a warning. Why does the cell see a double-stranded RNA as a warning, particularly a double-stranded RNA that the cell itself made? Okay. Well, the reason it sees it as a warning is because not all double-stranded RNAs are made by cells. Some viruses contain double-stranded RNA as part of their replication cycle. So if a cell gets infected by a virus that makes a double-stranded RNA, this warning signal makes very good sense. The cell says, hold on, we're going to stop this sucker right here in its tracks. So it stops the synthesis of cellular proteins that contain the sequence that's in here. That's what this thing does. That's the warning. If you stop a virus from being able to make its own proteins, the virus can't function. So this warning works really, really well. The cell has its own protection against an invading virus. Well, why does it make its own double-stranded RNAs? Solely for the purpose of regulating some of its own genes. It's odd. The cell goes to the trouble of making a messenger RNA, getting the messenger RNA to a ribosome, and then stopping the synthesis with something like this. Why do you suppose cells do that? People frowning. You should frown because it's puzzling. Why should a cell do that? Well, you don't know. I'll tell you. Okay? We don't completely know the answer. But one of the answers is probably this. When we look in our cells, we have messenger RNAs that we make. And those messenger RNAs that we make tend to hang around for a while. You might need the protein only an hour or so after the messenger RNA was made, and you wouldn't want it hanging around forever. So one thing you could do is you could destroy the messenger RNA. That doesn't happen real rapidly in our cells. Another way that you could stop it would be to do this and stop the synthesis of the protein that it codes for. And that appears to be one of the reasons why this system is powerful for cells. It allows the cell to stop the synthesis of any protein anytime that it wants. Okay? So this microRNA complex or siRNA complex, that's what they're both essentially for our purposes the same thing. These complexes allow the cell to stop synthesis of a specific protein, whether that's a viral protein or whether that's a cellular protein. That makes sense? It's Monday, right? I was telling people up front, my other class, everybody in the morning class just kind of sat there like this. And it's also week 5, and everybody's been studying for midterms. How many midterms this week? Okay, not very many. You guys should be wide awake. Or you're too tired to raise your hand to say we have midterms this week, right? Comment? Yeah. Yeah. It could. It very well could, yes. And that would allow it to just stop the synthesis of that protein. Kind of cool. What we'll see when we talk about gene expression is that cells have an amazing diversity of controls for determining how much of any given protein it makes and when it makes it. And this may be a factor in determining when a protein is made, or if a protein is made. It's a very powerful tool against viruses. However, it relies on the fact that it would only work against a virus that had a double-stranded RNA in the process. By the time this thing got geared up for a virus that wasn't double-stranded RNA, the virus probably would have killed the cell. Okay. Well, that's what I want to say about RNAs. And since I think I've exhausted your questions there, I'm going to start talking about replication of DNAs. Okay? Replication of DNAs. Everybody studied replication of DNAs, and you learned replication of DNAs. G codes for C, and A codes for T, etc. Okay? And that was something that was known very early on from the structure of DNA. When Watson and Crick and Franklin discovered the structure of DNA, it was quite clear that the complementary base pairs within the double-stranded duplex, one strand could specify enough information to make the other strand using the base pairing rules. A lot of details about how the replication actually occurred came in subsequent years. One of those was shown right here. This was a famous experiment that was done fairly early on by a couple of professors named Messelson and Stahl. Frank Stahl later was a professor at the University of Oregon before he retired. In this experiment, they wanted to answer the question, when a DNA molecule replicates, how does it replicate? Does something in the cell come along and copy both strands at the same time? Or does something come along in the cell and copy one strand and one strand? Well, if it copies both strands at the same time, you would see that if you started with something like this, you would have 2 old strands in one of the duplexes, and you would have 2 new strands in the other one of the duplexes. And so Messelson and Stahl says, well, we could answer that question fairly readily if we use different isotopes, so the one's heavier, and we could separate them by centrifugation. We won't worry about the details. But suffice it to say, when they did the experiment, what they discovered was this method, which is called the semi-conservative method of replication, was in fact what cells used. It meant that one strand ends up being, the parental strand ends up being in one of the duplexes along with a new strand that goes with it. And similarly, on the other duplex, one old strand and one new strand. That meant that the new strand was being made directly from each of the old strands. Okay? So the semi-conservative model of replication is the replication mechanism that all cells use. When we look at prokaryotic cells, that is bacterial cells, and we compare them to eukaryotic cells, we tend to see something very different in the overall process. As we will talk later, when I talk about the enzymes that are involved, the enzymes really aren't that different between prokaryotes and eukaryotes. However, the organization of the DNAs themselves, quite different. On the left, we see prokaryotic DNAs. Prokaryotic DNAs are circular. They're almost all circular. And replication starts at a single place called an origin, and then spreads out in both directions from there. You can see it spreading here, and you can see the new strands being synthesized, the new strand being the light blue colors there. Okay? When we contrast that with eukaryotic DNA replication, we see that eukaryotic DNAs are linear. They're not circular. And eukaryotic DNAs are much larger. I've already talked about how eukaryotic DNAs are about 1,000 times larger on average than prokaryotic DNAs. It turns out that instead of having one origin for the entire giant eukaryotic chromosome, that eukaryotic chromosomes have many more origins of replication. Okay? Many more origins of replication. Okay? Now, it turns out that there's something else that we'll talk about later that's unusual because of the way eukaryotes replicate their DNA. Eukaryotes have difficulty replicating the ends of the DNA. Every cell division of a eukaryotic DNA results in, and this may surprise you, loss of a little bit of the end of each eukaryotic DNA. It means that if you look in my cells and you compare them to your cells, I'm older than all of you in here, my chromosomes will actually be shorter than yours will. That's not good, by the way. Okay? My chromosomes will be shorter than yours will, because mine will have gone through more replications than your cells will have gone through, and consequently, I'll have less of my ends there. Okay? Prokaryotes don't have that problem. Prokaryotes, because they're circular, as we will see, are perfect. They get all the way through. They don't lose anything in the process. Well, let's talk about the actual synthesis. So the synthesis of DNA is catalyzed by an enzyme known as DNA polymerase, which I'm sure you probably know. And a DNA polymerase does several things. First of all, it binds to a DNA. It reads the sequence of one strand and puts the appropriate complementary base in on the other strand that it's making. A DNA polymerase will make the phosphodiester backbone of DNAs. That is, it catalyzes the synthesis of the bond between the phosphate and the sugar. Those are phosphodiester bonds. Okay? Now, let's take a little better look at the overall process than what this figure shows you. Okay? Let's look at what's going on. We see that replication of DNA occurs at a junction that we call a replication fork. A replication fork. And the replication fork involves, now remember we had a bidirectional replication, right? When we had the prokaryotes. So one fork is going this way, and the other fork is way over here. We're not even going to show it or think about it for the time being. Okay? So we've got a fork moving to the right. When we look at that fork moving to the right, we discover that this new strand that's being made on the top is being made in one single piece. It's called the leading strand. Okay? The strand that's on the bottom is not made in one piece. It's made in multiple pieces, and it's called the lagging strand. The pieces have names. George, Henry, Harold. No. The pieces have, the name of the pieces are called Okazaki fragments. Okay? Now you might wonder, why does the cell make these guys in pieces and it makes the top one in a single piece? And the answer has to do with the polarity of DNA. The polarity of DNA. You'll notice the top strand is going 5' to 3'. It's complementary to the 3' to 5' of the strand that it's copying, so they're anti-parallel. And that means that since the bottom parent strand is going 5' to 3' this way, that the only way that those guys can be paired is that way in the anti-parallel direction. Now we've got two things that come up here. One, DNA polymerases only work absolutely, it's one of the few absolutes, only work absolutely in the 5' to 3' direction. They can only synthesize DNA 5' to 3'. What's happening on the top strand? Well, there's the 5' end. It's got it made. It just keeps going and going and going and going like the ever-ready bunny, making 5' to 3'. It doesn't have to stop for anything. What does it take to synthesize in this direction? Well, imagine that I keep this guy going up a little further. I'm going to open up a new stretch of the bottom strand, right? That new stretch will be single-stranded, but because I can't make DNA 3' to 5', I can only make it 5' to 3'. It means that something out here has got to start and point it back that direction. So the DNA on the lagging strand gets made in individual pieces called Okazaki fragments. That is the reason we see leading strand being in one piece and lagging strand being in multiple pieces. Yes, Mariah. You're ahead of me, but I will talk about that. The Okazaki fragments need to be put together. Okay? So I want you to keep that in mind. We will have to put them together, but I want to save that for just a few minutes. Okay? We've got to put those pieces together. Now there's another thing that affects this. DNA polymerases have a very odd property, in my opinion. Okay? Very odd property that they have is that they can only extend an existing fragment. They can't start, if this were just a single strand down here, they can't start the single strand all by themselves. But if there's a piece that there's an end of, they can extend the end of that. So that's what this guy's doing. It's extending the end of something that's already been made. The question is then, how did it get started? If it can't start synthesis, how does a DNA strand get started? And for this, the cell has to use an odd trick. The odd trick it has to use is that cells use an enzyme called primase, P-R-I-M-A-S-E. And primase starts the DNA synthesis by putting down a small piece of nucleic acid where synthesis of the DNA polymerase is supposed to begin. What's the odd thing about it? The odd thing about it is primase makes RNA. Primase makes RNA. So the DNA polymerase grabs the end of that RNA and says, oh, you want me to start here? And then it starts and replicates and replicates and replicates and replicates. It means that for every one of these Okazaki fragments, every one of them started with an RNA made by primase. Primase is a busy enzyme. Very busy enzyme. But if you think primase is a busy enzyme, you ain't seen nothing. DNA polymerase is an incredibly busy enzyme. Okay? Yes? Primase puts down RNA, a small segment of RNA, and the end, the 3' end of that, is what the DNA polymerase adds to. Once it gets added to, everything is fine. Yes, sir?
Does it make the RNA or does it grab it from elsewhere?
It makes the RNA. Primase makes the RNA by copying a portion of the other strand, just like DNA would, like a DNA polymerase would. Yes, ma'am? You do have to get rid of the RNA. Okay, so Mariah's got two things ahead of me here. So we've got to get rid of the RNA, and we've got to attach the pieces. Okay? So I'm going to show you all the proteins involved in DNA replication in just a few minutes, and when I do, then we'll see the answer to her questions. But before I do that, I want to tell you something about the magic of a DNA polymerase. You see this going on right here at the replication fork? That's one DNA polymerase that's doing both of those at the same time. It works on this for a while, then it looks back and works on this. Then it works on this for a while, then it looks back and it works on this. It's looking both directions all the time. It's a remarkable thing. Okay? That's the first thing about DNA polymerase that's remarkable. But it's the second that'll blow your mind. DNA polymerase is moving along a replication fork at the rate of 1,000 base pairs per second. 1,000 base pairs per second. That's pretty hard to fathom. Okay? 1,000, can you imagine doing anything 1,000 times a second and getting it right? If you can type 60 words per minute, okay, each word being roughly 300 characters, each word being 500 characters, that's 300 characters per minute, maybe 5 characters per second, maybe, if you're a good typist. Okay? Polymerase is reading and typing at the rate of 1,000 every second. That's 200 times faster than you. And here's the clincher. Okay? It makes about 1 error in E. coli per every 10 million things it copies at the rate of 1,000 per second. DNA polymerase is pretty amazing. Okay? Roughly 1 error in E. coli for every 10 million bases it copies. Okay? And it's doing it at 1,000 of those per second. When we consider that an E. coli genome has about 6 million base pairs in it, it means it's making less than 1 error per every complete replication of that DNA. Pretty incredible. Questions on this before I move forward? Yes? Well, it's looking one at a time, but it can't go too far in one because it'll lose the other. So it's going here, back and forth at the rate of 1,000 a second. Okay? Imagine that you're a typist, right? And you're trying to type something, and you're reading this manuscript, then this one, then this one, then this one, then this one, and you're doing that at that rate. It just blows your mind. Yeah? Yes. All DNA replication systems have Okazaki fragments. At least all cellular DNA systems do. Yes. Okay. Now, let's see. Before I do that, let me jump around just for a moment. Let's look at the proteins, since Mariah's asked the question. Let's look at the proteins that are found at a replication fork. Okay. What's found at a replication fork? First of all, we have the lagging strand on the right. We have the leading strand on the left. The leading strand is moving upwards. The lagging strand is being replicated downwards. So they're going in opposite directions, just like before. The leading strand is in one piece. That's this solid purple line over here. The lagging strand is in multiple pieces. That's these multiple RNAs that we see over here. What else can we see? We can see, in the case of the lagging strand, this little red segment. And you see numerous red segments there. The red segments correspond to the RNA primer. Every time we have a DNA that gets started, it has an RNA primer that comes from primase. We can see a brand new one getting started right here, and it's going to come around here and replicate this direction. Okay? So primase is one of the proteins found at a replication fork. Here's DNA polymerase at the replication fork. Okay? What else do we have at a replication fork? There's something here called a sliding clamp. What the heck is a sliding clamp? Sliding clamp is a protein. It binds to the DNA polymerase, and it helps to hold the DNA polymerase onto the DNA. You see that little ring structure. We can think about it like my hand. My fingers wrapping around a strand of DNA. So if this guy here, my hand is now bound to the DNA polymerase, and this guy is holding onto the strand, then the DNA polymerase will be held to the strand. That sliding clamp turns out to appear in virtually every cellular replication system. Our cells have something very similar to the sliding clamp of E. coli. Very, very similar to the sliding clamp of E. coli. The sliding clamp is also called in E. coli, it's called a beta clamp. I'll call it by either term, beta clamp or sliding clamp. Okay? In human beings, this protein is called pCNA. Not that that really matters, but there's a name for you, pCNA. Okay, now what else is at the replication fork? Well, let's think about what has to happen at a replication fork. I said, well, this guy just goes along synthesizing in this direction, but of course, this is a duplex up here. How did the strands get apart? And how does the polymerase have a single strand to work on? Well, it turns out that something has to pull the strands apart for the DNA polymerase. There's a protein called helicase that is ahead of the polymerase that has the sole job of pulling the strands apart. That's all it does, is it pulls the strands apart. Now, helicase is another remarkable protein. You guys are getting tired of remarkable proteins, I can tell. But it's another remarkable protein. I'll tell you why. Polymerase was a remarkable protein at how fast it went and how accurate it was. Helicase has got to stay ahead of the DNA polymerase, because polymerase is only going to copy a single strand. It can't copy the duplex because we have semi-conservative replication. It's got to have single strand to copy. And the only way we get single strand is by pulling double strands apart. How fast does helicase have to work? Well, it's got to work at 1,000 base pairs a second at a minimum if it's going to stay ahead of the DNA polymerase. 1,000 base pairs per second corresponds to, it turns out there's about 10.5 base pairs per turn of DNA. That means every second there's 100 turns of DNA that have to get unwound. This thing has to spin at 100 turns per second to stay ahead of the polymerase, meaning that this helicase is spinning at 6,000 RPM. That's faster than your car. It's spinning at the rate of 6,000 RPM. Where does it get its energy from? ATP. ATP powers this sucker going at 6,000 RPM to pull apart the strands of a DNA duplex. Okay? The nanoscopic world is really remarkable. There's some other things on here. Here's something called SSBs. What are SSBs? SSBs are single-strand binding proteins. And they do as their name would suggest, they bind to single strands. Excuse me. Allergy season. Why does something bind to single strands? Well, a single strand is the most vulnerable type of DNA for a cell. Because if damage happens, there's no way of repairing that. It's gone. This whole thing would fall off. If I broke that right there, this whole piece would be lost, and the cell would be out of luck. The cell has single-strand binding protein to reduce the likelihood that this single strand is going to get damaged. It's protection. It's insurance. Gee, I'd sure hate if something happened to that strand there, you know what I mean? Right? It's protection. The cell has bought protection with single-stranded binding protein. Okay? What else do we have on here? We have something up here called a topoisomerase. At the very top, we've got topoisomerase. What is a topoisomerase? You remember the supercoiling thing I showed you the last time where we changed how many turns that we had? Right? And we saw that it all got all coiled up as a result of that. Imagine I'm pulling this sucker apart at 6,000 RPM. Do you suppose that the tension ahead of here might increase? The number of turns, base pairs per turn might change? Do you suppose it would get all kinked up? Do you suppose that would be a problem for the cell if it didn't get untangled? The answer to all those is yes. Okay? It would be a problem if all those didn't get untangled. And so to reduce that tension and go back to the more relaxed state, cells have enzymes called topoisomerases, and their job is to release the tension that gets built up by the helicase. The primary topoisomerase that's involved in DNA replication is called DNA gyrase. G-Y-R-A-S-E. DNA gyrase relieves the tension that's built up by the helicase during the process of replication. Okay, I've accounted for almost everything. There's two things down here I haven't accounted for. Okay. You'll notice down here it says DNA polymerase 1, and you go, what the hell is that? This says DNA polymerase here. What's a DNA polymerase doing down over here? Notice it's on the lagging strand. Okay? It's on the lagging strand. You don't see one over here on the leading strand. You just simply see something that says DNA polymerase. Well, let's clarify some things a little bit. First of all, this DNA polymerase has a name of its own. It's called DNA polymerase 3. DNA polymerase 3 makes the vast majority of the DNA in an E. coli cell. It makes the vast majority of the DNA in an E. coli cell. E. coli has about 5 DNA polymerases. The primary one for most of the replication is DNA polymerase 3 because of what it does at the replication fork. However, there are pieces of the DNA of a prokaryotic chromosome that are made by DNA polymerase 1. What is DNA polymerase 1? DNA polymerase 1 is very much like DNA polymerase 3. It makes phosphodiester bonds. It copies the other strand and puts in the right bases. But it also has another function that it performs. DNA polymerase 1 has, Gesundheit, you're like me, has an enzymatic activity that is not found in DNA polymerase 3. So they both make phosphodiester bonds. They both read the bases. Okay? But DNA polymerase 1 also has the ability to degrade DNA or RNA starting at the 5' end of a strand. It can degrade DNA or RNA starting at the 5' end. Remember the Okazaki fragments? Remember the pieces? And the very beginning was an RNA? DNA polymerase 1's ability to chew out that RNA allows it to remove the RNA, as Mariah wanted over here, so that the RNA doesn't appear in the final product, and the DNA polymerase part of it can fill in those little segments where the RNA was. So some of the DNA in the bacterial DNA come from polymerase 1. The vast majority comes from polymerase 3. And the last piece was what she asked about, well, how do the pieces get put back together? We filled in the gaps, but we haven't put them together. That takes another enzyme called DNA ligase. DNA ligase joins individual fragments together. Now, that's a lot of stuff. That's a lot of stuff to know there, okay? Look at all the different proteins. Look at that orientation. And this sucker's going along at 1,000 nucleotides a second. I feel very inadequate by comparison to that, okay? That's pretty remarkable. Questions on that? Yes, I see two. Mariah first. DNA gyrase is a topoisomerase. There are other topoisomerases, but DNA gyrase is one of those. Question? Same question. Yes? Good question. So this general scheme is true in eukaryotes and prokaryotes. Eukaryotes tend to have things with different names, and we're not going to worry about the names of eukaryotes. You don't need to know, for example, it's a pCNA. Okay. But they have the same general functions in eukaryotes and prokaryotes. And there are some differences, but it's way beyond the scope of this class to talk about that. Another question? Yeah. What's that? That's a good question, actually. I don't know the answer to that question, but my suspicion is that it would periodically slow down. Maybe I could think of an exam question right here in front of you guys to say, well, how is it that you might predict, based on what I've told you of enzymes here, where helicase might get slowed down? Oh, that's a very good observation. So GC base pairs are harder to pull apart. Maybe if it hits a rich section of GC, that might slow it down. It might. Yep. I can think of at least one other reason it might slow down. True, although usually if that's the case, the cell's in pretty deep doo-doo. But you're right. It could be another one. That wasn't what I was thinking of. What else? Oh, now you're really getting deep there. If the DNA changed handedness, which it can, then that would pose a topological concern. I would say that's possible. What if the topoisomerase doesn't relieve the tension? Okay, each of you get a point of credit. Okay, and that was the one I had in mind actually. Topoisomerase. What if the topoisomerase got blocked somehow? It turns out that some antibiotics work by inhibiting bacterial topoisomerase. If you inhibit topoisomerase, what's going to happen? Well, this guy's going to get really tangled at some point. Okay? And if it gets really tangled, the helicase hasn't anything else that it can do. It's like tying a knot in your DNA. The helicase is going to sit there and spin its wheels, and nothing's going to happen. Okay? That is an important consideration. Does that all make sense? Topoisomerase inhibition will generally result in cell death, yes. Yeah. There's one, in fact, I'm almost out of time, so I'll tell you a quick one, then we'll do a song. So there's ciprofloxacin. I think ciprofloxacin, yeah, is what it's called, right? Relatively new antibiotic. And it inhibits topoisomerases. Okay? One of the topoisomerases in E. coli has a single function. Think about the replication of that circle. When you get the circle done, you think you have two circles, right? But it turns out that when you replicate circles like that, you don't have two circles. You have this. And if you don't resolve that structure, what will happen is the cell will not be able to divide, and the cell will ultimately die. And one of the antibiotics that works like Cipro does the following. It inhibits that topoisomerase whose only function is to do this. If you stop that one function, you can kill bacteria. It says something pretty cool about what we can do with antibiotics, right? You stop that one function, you kill bacteria. Okay, more than you wanted to know there probably, so let's think about the weather, okay? I don't know about you guys, but I'm tired of the rain. I want you to sing along with me the following song. This is our life, folks. This is the truth. It's got birds and everything. All right, think dry thoughts, guys.